Welcome to Let's Get to the Mark. It's just a quick sound check. So, yeah, I'm going through 4 o'clock every day through the Easter holidays. So I'm going to be going through GCSE Science. I'm going to throw in a few A-level ones. Might start later this week, maybe next week, with AS and A-level for chemistry and biology. I'm working on biology at the moment, and so this is organisation, which is Unit 2. Hello, how's it going? Um, just a quick sound check. Can you hear me? Evo 1163. Can you hear me? Like I say, I'm using some crusty little laptop here, so I don't even know whether anybody can hear me. But yeah, let's go. Let's get on. Yes, you can. Am I clear and everything, or am I a little bit quiet? Hopefully, I'm all right. Okay. As I say, I haven't got any fancy stuff going on. So with my lives and some of my previous live, I have little video clips and things like that. It's going to be nothing like that at the moment. We're just going through the stuff. So nice one. Thank you, Kivo. 1163. Let's go. Let's get on with the live. So I'm looking at AQA Biology Combined Science Triple Science Unit 2, which is organization. This is coming up on the 12th of May in your AQA Biology exam or your Combined Science exam. And so just some basics here. Just a little screenshot from the CGP education purposes, obviously, borrowed this. But essentially, just knowing that cells come together, you get cells and they work together in a group, and the example they want you to know in the AQA syllabus is epithelial tissue. Now, this is the tissue that covers the inside of the stomach, but it's also the tissue that covers the inner line or covers many of your organs. So you've got it in your lungs, your alveoli, the cells around the alveoli, etc. Any cells, the cells lining your intestines, those are epithelial cells as well, the ones with villi on them. So epithelial cells are just a group of cells working together to cover an organ or the surface of an organ. So it's just knowing what a tissue is, that it's a group of cells doing the same job. And then an organ is a group of different tissues all working for the same job. And the example they give you here is knowing that muscular tissue in your stomach churns the food, glandular tissue makes digestive juices like protease, and then epithelial tissue covers the lining of the stomach. Anywho, um, moving on, you will need to know, and this is probably make up 1% of your biology paper. One, if you're doing triple or separate science, as they call it, it's going to be out of 100 marks. If you're doing combined, it's going to be out of like 70 marks or something. So it's a few marks less. But basically, you could well be asked to name an organ. So just make sure you know what A, B, C, uh, D, E, F, G, and H are. You have to know the names of all these parts of the digestive system. So make sure you know them. And I would say common things they could ask you is like, how is protein digested in the stomach? That could be 3 to 4 marks. Now they could ask that in context. 1 year in IGCSE biology, they said, explain how this piece of chicken was digested in the stomach of a rat. And that threw loads of kids off. They come out, oh, sir, I didn't learn about how chicken's digested in a rat's stomach. Man, we never got taught that by our teachers. But really what they're asking you there is, how is protein digested in the stomach? So what role does the stomach play in protein digestion? That's worth about 3 to 4 marks. So make sure you know that. If you don't know that, put that on a revision card, put the answers on the back. I'll go through it in a second. You also need to know, what role does bile play in digestion? So bile has two jobs in digestion. You might know the names of those jobs. And it's probably about 4 marks altogether. So you might be asked about one of those jobs, which is 2 marks, or you might be asked about both, which is 4 marks. So make sure you know the roles that bile plays in digestion and protein in the stomach. So I'll run through the name of the organs, although you might be able to do that yourself. So do it in your head as you go along, and you might be able to do it yourself. Um, so you can do it in your head as you go along. So A, somewhere up here, normally around the jaw, are the salivary glands. Obviously, A is pointing towards the mouth, but you need to be aware of the salivary glands, and they make amylase. Amylase breaks down starch into sugars. We then have the esophagus. One simple job for the esophagus is to deliver food to the stomach. That's all it is. Now, it is a muscular tube, so it contracts and relaxes involuntarily, even when you're asleep, doing that. And we call that peristalsis. You don't need to know that for AQA. If you were doing IGCSE, you'd need to know about peristalsis. Now, the food is moved from the esophagus into the stomach. which is part C. So if you knew that C was the stomach, well done. And how is protein digested in the stomach? You need to know that. B is the liver. The liver has like 500 jobs in your body. It's a super important organ. It plays one significant role in digestion, and that is that it produces bile. It makes this stuff bile, and bile is stored round about here, what is supposed to be G, in something called the gallbladder, a little organ that connects to the liver, and the bile is stored in that and then secreted from that into your small intestine. Now, E is the large intestine, F, we got the pancreas there, and D is the small intestine. The small intestine is where small food molecules like amino acids and sugars transfer into the blood. And then you've got the rectum here where you store your waste, your feces, and then the anus, the hole at the end of your digestive system where you excrete. Now, um, what do we need to know about protein digestion? The reason I mentioned protein digestion, protein, so you're thinking fish, chicken, eggs, beans, lentils, nuts, these things that are high in protein. The majority of the protein is broken down in your stomach. Okay, and how does that happen? Well, right here. So protein digested, 4 marks. You need to talk about protease, the enzyme. It's made by epithelial tissue or the glandular tissue actually in the stomach, and it's secreted into the stomach. You probably won't need to talk about that. You just need to mention these bold things. Protease or sin is the specific name of a protease. Breaks down protein into amino acids, and that's 2 marks. It loves acidic conditions, pH 2, which is why the stomach has hydrochloric acid in it. So make sure you know that for protein digestion, you can get 4 marks. It might only be a 3 marker, but you want to know the 4 things. So protease enzyme, called pepsin, breaks down protein into amino acids. So you want to say the product that they get produced into. Protease or the pepsin enzyme likes pH 2, acidic environment. It's optimum for the digestion of protein. And it's the hydrochloric acid that creates that acidic environment. Any questions about that protein digestion, you can put up in the chat and I'll answer them. What else? What else? What else? Yeah. Hydrochloric acid also kills bacteria. It's like primary defense of your body to prevent. It's a chemical defense. So you do need to know that when you come to Unit 4 in Paper 1. Hydrochloric acid, chemical defense, it does kill bacteria. You might pick up a mark for that. But specifically on digestion, the hydrochloric acid is to create an acidic environment, which is the optimum pH for the protease enzyme. Let me have a little look. What's the hardest question they could ask? What, on digestion or just generally in biology? Um, I'm going to go through the whole of Unit 2. On this topic, um, depends what you find difficult. Some people find the lifestyle factors or environmental factors, things to do with the heart. Probably a lot of people traditionally have issues with the heart and with how the heart works, really. And so I'm going to go through that. Um, that's probably what people find difficulty with, but I'm going to go through everything. So make sure you know about bile, guys. Bile. What does bile do in digestion? Let's have a look at that separately. So bile is made here in the liver and stored in the gallbladder, and bile neutralizes the hydrochloric acid. Remember, the food's leaving the stomach. You've eaten your chicken burger. That chicken burger, or if you're a vegetarian, you've eaten your nuts or your whatever, that's leaving your stomach and going into the small intestine. Now it's coated in acid. Lipase and amylase enzyme do not like acidic environments. It denatures their active site. So we need to get rid of the acid. And so bile is squirted onto the food and neutralizes the acid so that you have an alkaline pH, which is optimum for lipase activity. Lipase is the enzyme that breaks down lipids and turns them into fatty acids and glycerol. So if you get a question on fat digestion, it could be worth 4 marks if it's an explain. Bile neutralizes the acid, creates an alkaline pH optimum for lipase. Lipase breaks down lipids into fatty acids and glycerol. Bile also has another role. So you could get a fat 6 marker on digestion. Um, Bile emulsifies fats. That means breaks them into little pieces, which increases their surface area so that they can be broken down more rapidly by lipase enzymes. Um, yes, I'm going through the triple dipple stuff as well. I'll check the spec, make sure I've covered everything as well. So this is all triple as well. Make sure you know the food tests. I'd say if you're asking me on this topic, now these are easy peasy food tests. But I guarantee you 90% of students, even those on a level 9, flop one of these food tests. So make sure you know the food test, whack them on a revision card and learn them off by heart. Because food tests come up, they're only worth a few marks. There has been some 6 markers on food tests in both the triple and the combined. So make sure you know the food test. Do you know what the test for sugars is? What is the test for glucose? Potentially is 3 marks. You could put in the chat if you know what the test for sugars is. So feel free to jump on the chat and bust out the test for sugars. So you're looking at the 3 things you want to talk about is Benedict solution, which is blue, and the color change. Heat Benedict's lovely. And if you don't say heat or warm, you lose a mark. So Benedict's is the only one, the sugar test is the only one that has 3 bullet points. So you just want warm or heat. Obviously, it's better to use a water bath than a Bunsen burner, but whatever. But heat, Benedict's, and it goes red. Now, technically, it's semi-quantitative, which means it gives us a slight indication of how much is there. The other food tests do not give you an indication of how much is there. This one is semi-quantitative. It doesn't give you a number, doesn't tell you like, oh, you got 5% sugar, but it will go green, yellow, orange, red kind of thing. So it works its way from blue across the rainbow to red, red meaning there's lots of sugar, yellow, green, or whatever, meaning there's just a little bit of sugar. So it's quantitative, really a qualitative test. Um, but yeah, protein. People get these two mixed up, so they can never remember if it's Benedict's. What's the one for protein? Anyone know the protein test? So you've got to use a different solution, but it begins with a B. So this is why I would say lots of students flop on the food tests. So the test for protein is Biuret's, and there's only 2 things you need to say with Biuret's because you don't have to heat it. Just Biuret's goes purple. Now, do you have to know the color of Benedict and Biuret's? They're both blue. Not really. I know people learn blue to red, blue to purple, but thing they're marking is what is the positive result? What is the color? What does it go to? So you really need to know that Benedict's turns red, Violet's turns purple. Yeah, and it turns purple. And what I like is protein begins with a P. And if you can get it in your head, P for protein, purple, you're going to always pick up a mark. So if you're like, what's the test for protein? You're like, is it Benedict's? Is it Violet's? At least you can go, well, protein begins with a P, it's purple. So that's the kind of thing. Starch, starch test. Do you know what chemical you need for the starch test? Now, lipids isn't actually on your syllabus. So all I'm going to say is half the kids I teach are like, oh, so we need to know about Sudan 3 and the red layer and the floating layer and all of that. Look, Sudan 3's lipids test is not on your syllabus. That doesn't mean they won't mention Sudan 3. But I've noticed specifically, it doesn't mean they won't mention it in like a matchup or something and it's the odd one out. It might be mentioned in a question, but I don't think they can actually ask you what is the test for lipid, like directly up. I don't think it's on your syllabus, the lipids. Iodine goes black, that's right. So iodine is orange. It looks like henna, the kind of thing you get like wedding tattoos and stuff with. It's like orange in color, brown orange, and it goes blue black. And lipids, you can shake with ethanol and it goes cloudy, or you can use the Sudan 3 test where you get like a red layer that floats. Um, it separates into 2 layers and one of them's red, but I actually don't think it's on your spec, and I'll show you that at some point. So, yeah, what warm Benedict solution, blue or red? These are the answers basically. Also, they asked a 6 marker a couple of years ago, and it was you might have even done this 6 marker. And it was somebody's got some food, describe the test for carbohydrates, 6 marks in the food. And I think some students for a required practical, and we had the opposite. What was this? The opposite. Starch disappearing, going from black to orange instead of orange to black. I'll explain that, Kivo. The required practical. I was going to do all the required practicals on another day. It's not that it's opposite, but yeah, you're absolutely right. That is what happens in the required practical. I'll explain that in 2 seconds. I will do a separate thing on all the required practicals. I'll do that as a separate hour. Um, but I'll explain that in 2 seconds. There was a 6 marker which said describe how you test for the presence of carbohydrates. And I think students got a little flopped on this because it might have said protein and carbohydrates. So they did the biurets, got the 2 marks out of 6 for the biurets. They did the starch, orange to black, and they got the 2 marks for that. But they forgot that glucose is a carbohydrate. So sugar and starch, glycogen, and cellulose are all carbohydrates. Anything that ends in OSE is a carbohydrate. We might say, well, starch doesn't end in OSE. Well, its real name is amylose. It's broken down by amylase. Glucose, anything ending in OSE, basically, fructose, sucrose, glucose is a carbohydrate. So if they ask you for the tests for carbohydrate, they want you to bust out the sugar test and the starch test. Anywho, let's move on. We don't want to be hit all day. Yeah, now, Kivo, you mentioned about a question for the required practical was the opposite. I'll do that on another day, but essentially, what you're doing with that required practical is the enzyme required practical. And what we look at there is starch and how quickly, how temperature affects starch breakdown. Now, if you think about it, um, starch being present is black. So when you start with starch before it's been broken down, it will have already turned from orange to black. So your iodine is always orange, but you add it to the starch and it goes black straight away. So you start off with it as black. And then when the enzyme amylase breaks the starch down, it will no longer be black. It will just look orange. So say I have my solution which has starch in it and I add my amylase and I start my timer, it's going to be black at first because starch is in there. After the enzyme breaks it down, say 10 minutes later, if I tested it with iodine, it wouldn't be black. The iodine will be orange and stay orange. And so that's why it appears like it's going black to orange, really, because we're breaking the starch down into sugar. So but I'll do that in a separate for practical. And you can change the temperature for that or the concentration of the enzyme or the pH in that practical. That's an enzyme core practical. That's probably the person who asked me what's the hardest thing they could ask you. I'd probably say the core practical, the triple core practical enzyme thing. Right. Make sure you know these parts. So do you know A, B, C, and D? You can just test yourself in your head. I'm just gonna have some coffee. So I would say make sure you know what A, B, C, and D are. A is your trachea. You just need to know it has rings of cartilage. B is your bronchi, also has rings of cartilage. Notice these tubes, C, these are the small tubes. These are bronchioles or bronchioles, if you like it to sound Italian or whatever. But they don't have rings of cartilage. They don't really need the strength and the support. They're deep inside your lung. And then D is your alveoli. Good. And alveoli is the site of diffusion. Nice. Trachea, bronchi, bronchioles, alveoli. Spot on. Well done. Boom. Uh, yeah, I don't have all my sound effects, so I'll just do the live sound effects. Now, you do also need to know 3 things that all exchange surfaces have. You've got exchange surfaces throughout your body. We're big, large, multicellular organisms. We're not like bacteria, single celled. Bacteria can just let oxygen diffuse into their body because they're just a single cell. We're made of millions of cells. So we have to get that oxygen, and a cell will die in a couple of minutes without oxygen. We have to get that oxygen and get it down to our toes. So we have these exchange surfaces so that we can rapidly shift stuff around our body. You've got them in your intestines with your villi. You've got them in your lungs with your alveoli. So you've got them in several places, these exchange surfaces. You have them in your kidney where the blood goes through the kidney, needs to swap substances. So all exchange surfaces need 3 things to make them good. Plants have them too. Think about the leaf of a plant, um, the roots of a plant. What should all exchange surfaces have? These are surfaces of a living thing that exchange substances with the environment. What should they have? What is the type of tissue in plants that contains stem cells? Yeah, so I did that in the first slide. That's meristem tissue, and that's found at the tip of the root and shoot, Kivo 1163. That's found at the tip of the shoot and the tip of the root. You can clone a plant from that. It's called meristem tissue. That's like a one marker they could ask about. So good that you know that. Plants keep their stem cells. They're almost like embryonic stem cells. So they keep them all the way through their adult life. Of course, soon as we are go from the embryo stage, we no longer have, once we become a fetus, we don't really have any embryo stem cells left. So with a plant, you can chop a branch off, it will grow a new branch. You chop our arm off, we can't grow a new arm. We don't have those kind of like stem cells that can become anything. Um, the three things that all exchange surfaces need to have are a large surface area, hence why the alveoli have folds in them, and why we have millions of alveoli, because that creates a large surface area in the lungs. In your intestines, you have villi, which are further folded into microvilli, two separate points. Villi give a large surface area, microvilli further give a large surface area. So all exchange surfaces, large surface area. The leaves of plants are exchanging material with the environment, so they have a large surface area. So all exchange surfaces need a large surface area. That's the first thing. Second thing, they need thin walls, a thin diffusion pathway, short diffusion pathway, or a short distance for diffusion. So what do we mean by thin walls? Look at this alveoli wall. It's made of cells. In fact, this whole bit here is the wall of the alveoli. This here, this is the wall of the alveoli. Notice only one cell thick. So that means it's not going to be an issue for oxygen to how many cells is oxygen going through? Well, it's going through the one cell of the alveoli, and then it's got to cross the one cell. Notice the capillary is one cell thick. So it's only going to cross two cells in distance, the oxygen, to get to its destination, which is the red blood cells. So essentially, we say that's a short diffusion pathway. Don't make the mistake of saying it has a thin cell wall. You lose the mark for that because animals, and this alveoli are in animals, they don't have cell walls. So we're saying the wall of the structure, the wall of the tissue there, the alveoli wall is thin. The wall of the capillary is thin, not the cell wall because they don't have cell walls. Anywho, you want thin walls, large surface area, and the third thing is a good blood supply. Yeah, that's right. That's right, cheese. We need thin walls for a shorter diffusion time or shorter diffusion distance. And we also need a good blood supply. So these are the kind of answers. And if you don't know any of these, I suggest you write them on a revision card. Coffee done. Right. Thin walls. So the description is this. If you're asked for 3 marks, describe how the lungs make a good surface area. You'd say, oh, the alveoli have thin walls. There's a large blood supply and they have, what is that? Large surface area, right? If you're asked to describe and explain, then you have to give the reason why. So thin walls make a short diffusion distance, pathway, whatever. Extensive blood supply maintains the concentration gradient and large surface area, rapid diffusion. Don't say thin cell wall, only plants. And don't say that plants have a good blood supply. If you're doing how is the leaf got a good surface area? Yes, it's got thin walls and it has a large surface area, but don't say good blood supply because plants don't have blood. So, any questions before I move on? Feel free to ask any questions in the chat. Thank you. Um, yeah, let's move on. Yeah, I recommend if you grab a pen and some paper when I do the lives, you can just make note. Don't make notes of the things you know. So, like one of the tendencies we have as a student is to make notes on things we know because it kind of gives us a nice cuddly hug. So if I'm always revising, oh yeah, I know the structure of a cell really well. The nucleus contains DNA, and if you know that, you don't need to make a note on it. Like just make a couple of revision cards or some paper and just make notes on the things you don't know. So if you're like, oh, I didn't know that. I knew about the thin walls, but I didn't know that, and make a note on that. And that means you can go over the things that you don't know. Would I get a mark if I said one cell thick? Yes, but you will also get the mark for thin walls. My only danger, and 1 cell thick is even better. The only danger with it is that sometimes people go, "Oh, the cell wall is only 1 cell thick," and they just say the word cell wall. So it's really like this thing here is a capillary, and it's the wall of the capillary is made of cells, and the wall of the capillary is 1 cell thick. There's a tendency to go, oh, the capillary's cell wall is 1 cell thick. And then if you mention cell wall, they don't have a cell wall. These are all animal cells. It's a tissue, the capillary. Look, it's made of all these cells here, all doing the same job, which is, yeah. Oh, right. Moving on, we have, I really explained this one. If you can just remember this one before I move on, if you can just remember that having a large blood supply maintains the concentration gradient, you'll find. What do I mean by maintain the concentration gradient? Let me just show you guys. All right. Oi, where's the old pen? Right. Let's imagine these blob are oxygen molecules. Right. This is oxygen in the air. You're going to breathe in nitrogen and whatever else is in the air as well. But the oxygen is attracted to hemoglobin in the blood cells. So look, it crosses over. That is called diffusion. We're going from a high concentration to a low concentration. What happens if all of this goes over here and now I've got all this oxygen in these red blood cells and there's not a lot of oxygen in here? Which way is the diffusion going to go? Well, it's going to start going backwards. We don't want that. What, the oxygen leaving our blood and going into our alveoli so that we breathe it out? We don't want that. So in order to maintain, so it is always high in here and low in the blood, what have we got? Well, the blood by its very nature, it's like a river. It's moving like a river with boats. So these are moving. They're constantly, oh, Sugar Ray, Leonard. Right, what am I doing? Oh no, if I try to do that, it's trying to get too funky. I was going to try and select them and move them. But you know what I mean? They're being whisked away like that. They're being taken away. And so because of that, you will always have low oxygen here because these blood cells are moving the oxygen away. You'll always have higher oxygen in the alveoli than outside. And the way you just don't need explaining that's difficult, obviously. Watch me try to explain it. But you just need to say it maintains a steep concentration gradient or maintains a high concentration gradient. So it's always high there and low there. Right. Anyway, let me move on. Right. Go. Oh, sugar. What is that mean? No. Oh dear. It's all competes on. Right. Right, yeah. So on your revision card, TiVo 1163, what I recommend on revision cards, like some people just put the content. I think it's much better to put a question. And the reason is you can sit there watching Netflix, doing whatever you're doing, watching Disney, whatever you're watching in the evening, and you can have a stack of revision cards for like say B2. And you can just write B2 in the top left-hand corner, right-hand corner, whatever corner you choose, so that you know when they all get mixed up on dashed on your bedroom floor, you can put them together again into the right units. And so if you're making revision cards for B2, you could just be like, oh, how are the lungs adapted for gas exchange? Question mark on the front. Now, if you want to be efficient and have less cards, you could also put another question underneath. How are the villi adapted, or how are the small intestines adapted for gas, for exchange of substances, not for gas exchange? That's the alveoli. The reason is you'll see the similarities. Then you flip over and you write your answer. Now, how are the lungs adapted? You'd say, oh, they've got millions of alveoli, large surface area, thin walls, and a good blood supply. And if you want, you could write the explanation as well, i.e., thin wall, short diffusion pathway, good blood supply, maintains the concentration gradient, millions of alveoli, large surface area. Now, for the villi, for the small intestine, you write the same thing, but it's just adapted slightly. So large surface area is from the millions of villi, finger-like structures in our small intestines, and microvilli. So either of them, they're separate marking points, but you have villi and then further folded into microvilli to give an even larger surface area. And then also the intestines have a good blood supply. to whisk away the glucose. So you've always high concentration to low. It's always going from the intestines into the blood. And then they also have thin walls. The epithelial or epithelium tissue of the small intestine is thin, one cell thick, again, just like the alveoli. So very similar answers, and you could have them on the back. And that's what I would do with all my revision cards. I put some questions. So then you're just going through, time you get to the exam, you've got it drilled. Like for this one, I'd be like, okay, state the difference. I'd be like, describe the difference between a vein and an artery, and then describe the difference between a vein and an artery and a capillary. So you could have two separate questions. And then you're just going to go, which one, by the way, which one of these do you think is the artery? Which one is the vein? And which one is the capillary? F, G, or H? Which is which? Yeah, that's for the lungs, just for the lungs. But you can apply that same knowledge to the intestines. You just change the word alveoli to villi. The intestines, it's the same answer, but instead of alveoli, the intestines have villi and microvilli. Identical answer. So you could write identical answer, just change intestines to microvilli. Just change alveoli to microvilli, sorry. Yeah, and then you should be able to recognize what's F, G, and H here. You've got three blood vessels in your body at GCSE. We don't tell you about the other ones. We've got artery, capillary, and vein, which is which. So you've got one cell thick, that's going to be the capillary. H is a capillary. Notice it's the smallest. A capillary is about the size of one of your hairs on your head. And if you look in your eyeball, if you look in your eye, the white of your eye in the mirror, you'll find these red lines. Those are capillaries. They're very, very thin. But relative, i.e., compared to their overall size, their lumen's actually quite big. Look at the size of the space in them, even though they're only the size of a hair. Now, your veins and arteries are as thick as some of your veins and arteries are as thick as your finger. Your biggest ones in your body are as thick as a finger. So your artery has the smallest lumen because the blood has to be squirted at a high pace to go from your heart to your toes and deliver oxygen. So it has to be at high pressure, and it has the thicker wall. The wall is made of elastic and muscle, and the vein has the large lumen because it's at a lower pressure. It's already delivered its oxygen. Now it's just working its way back up your body um, to the heart to get repressurized. That's absolutely right. Yeah, the vein is F. Now the vein both have elastic and muscle. The capillary has no muscle and no elastic. It's just got an endothelium, one cell there. The there's elastic and muscle in both, but notice they're thinner in the vein. Now, elastic and muscle tissue have two different jobs in the blood vessels, but it's a little bit advanced. Elastic is for recoil. So the as blood goes past, can flex a little bit like that, right? Spring gives it a bit of springiness. And you've got the muscle you can say is for strength or to smooth the flow. Some people say the muscle is for smoothing the flow of the blood. So elastic is for recoil and the muscle is to smooth the flow of the blood. Anywho, um, yeah, cool. Let's move on. Yes, you need to know the artery has a small lumen relative to the large lumen for the vein. Now, why do I use the word relative? Well, I could have an artery that's, say, as thick as my little finger here, and that's got a small, but then I could have a vein that's even thinner. I don't know. Right? Or my artery could be as fat as my thumb, and one of my veins could be thin as that. So it's not that always, it's relative to their overall size. The artery has a small lumen. So you can just say at GCSE, we can just say the artery has a smaller lumen than the vein. And the reason is because of the high blood pressure. So you've got to link lumen size to blood pressure. So that's a really key link. The size of the lumen is linked to the blood pressure. So the artery has high blood pressure because it has a small lumen. The vein has a low blood pressure because it has a big lumen. Okay, so they're linked together, the pressures and the size. Think about a water bottle. You get your Evian bottle and you're squirting your mate in the summer with some water. You squeeze it, you're making a small lumen. The water shoots out. So the artery is trying to shoot the blood And so it needs a small lumen to create high pressure. Cool. Let's move it on. So you've got the heart, and you need to be able to answer these questions for the heart. What is the function? What the hell? Oh, my days. You need to be able to answer these questions. Make sure you can answer these. I'll just talk you through the heart. I have got some questions. I'm just kind of talking. Um, yeah. Yeah, the outer layer has to be tough, so it's got connective tissue, collagen fibers, that type of stuff. Now your deoxygenated blood enters the right atrium and it's then pumped down here. The right atrium contracts. Notice these gray bits are muscle. It's pumped into the right ventricle and then up here to the lungs. So it's being pumped. Oh my days. Check out that handwriting with the mouse. Not good. So it's pumped up to the lungs. Now, at the exact same time, Pete, these are not happening separately. They happen at the same time. The same time that blue blood comes into the right atrium. I shouldn't say blue blood. I should say deoxygenated blood, but we always traditionally draw it as blue and red. The exact same time this is happening, this is happening. Red blood is entering the left atrium, or oxygenated blood is entering the left atrium. You won't get marks for saying red or blue. And then at the same time that this ventricle squeezes, this one squeezes. This time the blood comes up here, out through here. And here's what confuses students. Sometimes in diagrams, they'll show it. It comes down round the back and off to the body. So it will sometimes come off and then go behind the heart. And that is the aorta. Um, You need to know about the pacemaker rather than the sinoatrial node. You don't need to know about the atrial node. You're doing A level, you've got to know about the SA node and you've got to know about the AVN, the atrioventricular node, and the sinoatrial node, the SAN. You don't really have to know about that at GCSE. At GCSE, you just need to know that we have a pacemaker here because your atriums are going boom, and then your ventricles are going boom, and they're doing it at a different time, right? Boom, 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 boom. There has to be a gap in between. That's pumping, those doors are open, that pumps, those doors need to be shut. We have these things called valves, which I've put here what are valves for. Valves prevent the backflow of blood. So once I'm squeezing this up to the lungs and this round to the body, I need to make sure these doors are shut. Otherwise, the blood's going to go back the way it came and not up through here. And so we need a pacemaker to coordinate the contractions. So the pacemaker keeps the rhythm of the heartbeat. And you just need to say it controls the rhythm of the heartbeat. It fires out an electrical nervous impulse through the cells and causes contraction. This is the body, by the way. This one's off to the body. Now, you could also be asked, you might be asked, why is it called a double pump? Or why do we call it a double circulatory system? So why is it a double pump? Why is it a double circulatory system? Your answer to that, if it's 2 marks, if it's 1 mark, your answer is the right side of the heart pumps blood to the lungs. The left side of the heart pumps blood to the body. So there's two circles going on like a figure, like an eight, like you're writing an eight. There's two circles going on at the same time. So it's called double circle, double circulatory. Now, the right side takes blood to the lungs, the left side to the body. If it's 1 mark, that's all you need to say. If it's 2 marks, you need to say the right side pumps deoxygenated blood to the lungs. The left side pumps oxygenated blood to the body. So it depends on how many marks. This is AQA Biology. So this is GCSE. So this is the exam you take at 16, and this is in the UK. Systole and diastole, Kivo 1163. It depends if you're doing GCSE or A level. Like this is my in the title somewhere on this, it should say GCSE revision. If I sorry, it should say GCSE revision. I will be doing A level and AS biology revision where you got to know a lot more detail than this. Um, Yeah, the fish do have a single circulatory system, Ishal. Yeah, fish have a single circulatory system. So fish, it's a less efficient circulatory system. It's not as good as our circulatory system. And they pump their blood to the lungs. They don't have lungs, they have gills. There are some fish with lungs, but never mind. Most fish have gills. 99.99999% of fish have gills. They pump their blood to the gills, which is their lungs, that picks up oxygen, and then it goes to the body. It doesn't come back into their heart. So they have a single circle. It goes heart, gills, body, heart, gills, body. Ours is a double. It goes heart, body, heart, lungs, heart, body. So you say heart twice. So it's a double circulatory system. The reason is, in order to get the force or the pressure, when our blood goes to the lungs, it loses energy, it loses force, it loses pressure. It won't make it around the body. So it's got to dip back into the heart. It's only 10 centimeters, 20 centimeters from the lungs. So it dip back into the heart to get re-squeezed. Now, make sure um, um, we can take a break. No, GCSE PE links a lot with this. Yeah, actually, Sanders, thanks. No problem. Yeah, cool, cool, cool. Look, GCSE, what am I going to say? Oi. You can always pause the video, have a little break, come back, and you'll always be able to rewind. This will be up on YouTube for a while. So, thing is, so you've got to be able to answer these. What are valves for? So valves stop the backflow of blood. Why is this wall thicker than the right? What would be your answer? Think about their destination. And that would be like 3 marks or 4 marks. Four things you got to say. Why is the left side, left ventricle thicker than the right ventricle? Think about where they're pumping. Why would you need a thicker wall? Oh, this session is going to end. I'm only doing unit two. Session's going to end in about 20 minutes, and then it'll be done. So I probably won't take a break now. I'll just smash through, and then in about 20 minutes it'll be done, and then I'll do another one tomorrow. So okay, yeah. So how 4 marks? What we got to say? Why 4 marks? Okay, I might be overdoing the 4 marks, right? I'm saying there's four separate points you need to get in your head for this. So I'll bash this on a revision card. Maybe not bash it, but write it on a revision card. Why is the left ventricle thicker than the right? So we're going to go, what is it made of? What tissue is your heart made of? Muscle. So I want to say it has more muscle. 1 mark. What does a muscle do? What word do we link with muscle? So, Sparky, yeah, it has to pump the blood everywhere in the body. Now, we need to get more technical, Sparky. You're absolutely right, but we need to get more technical. So, more muscle. Oh yeah, I'll come to them. A, B, C, D. I'll do them in 2 secs. So, more muscle. Why? What does the more muscle do? What all muscles do? There's a word you link with muscles. It's the word contract. So we want to say more muscle, more contraction, so the muscle can contract, and with what? Contract with stronger or greater force. So it contracts with a greater force. Why? Because it's sending the blood. Now, if we say it sends the blood everywhere, they might not give us the mark. We need to tell the examiner we know that the body is a longer distance than the lungs. So we have to use the word further or greater distance. It's all very technical to do. Biology is a very language based subject. So we need to say more muscle, greater contraction creates, you can use the word force or pressure, creates a greater force or greater pressure to transport the blood a longer distance. So four separate points you want to smash down when you do it. Okay. So blood is pumped at a higher pressure. Yeah. So stronger muscle, bigger contraction to create higher pressure to push the blood further. You must use the word further or greater distance. Okay. Very, very important that you stress that. Now, I could ask you, why is the right atrium smaller? Look at the size of this here. This is smaller than this. We've got the same answer, just different destinations. The right atrium is only pumping blood 2 centimeters to the right ventricle. It only needs little muscle for a small contraction and small pressure. The right ventricle in the blood like to the lungs a further distance, so needs thicker walls than the right atrium. So you get the idea. All of the chambers have different size walls depending on how far they're pumping the blood. We need to use the word distance, the word force or pressure, and contraction when we talk about a muscle. So in biology, the moment you're writing muscle, you got to be thinking about contraction normally. Normally, it's a word linking game. So, oh, hold on a minute. Yeah, deoxygenated blood. Now, this is key cheat code. This is a cheat code for the heart. This answer. Any time, right? I'm going to go here. Here's the answer. If you have a something wrong with your heart, so on a revision card, you want to write anything wrong with the heart. Now, that could be a faulty valve. They give you questions where they say, oh, this person was born with a faulty valve. This is a valve. It stops the blood going backwards. Or they say, that person was born with a hole in the heart. A hole in the heart means the blood in the left and right ventricle mix. So deoxygenated and oxygenated mix. Or if you have a faulty valve, then you're going to get less efficient blood pumping, right? Because some of the blood is going to come back up here when it was supposed to go up there. So anytime there's a fault, you just need to know these 4 key words or terms. You're going to get less blood to the cells or less oxygen to the cells, less respiration or even better aerobic respiration. So less energy released. So cells will die. And that is the problem. Anything that's wrong with somebody's heart results in that. It doesn't matter what's wrong with it. Anything wrong with your heart results in that. Less oxygen to cells, less respiration, less energy released, cells die. So you just need to memorize that as any fault with the heart is going to happen. Yeah. And the way to cure that is stents, Sadiqa. You are right. Stents, or what's the drug you can take? There's a drug you can take instead of having a stent fitted. So you do have to know about that in AQA. I'm going to rinse through that now, or just smash through that. So statins, thank you, Ishal. Statins, yeah, absolutely. So, coronary heart disease. By the way, guys, somebody did ask me what is A, B, C, and D. If you don't know what they are, you can sketch this out on a revision card. Write down A, B, C, D. Put that on the front. A, B, C, D, and then write what are these, and then on the back. It doesn't even matter if this doesn't look very good. Just make a little crummy heart drawing, and then just have A, B, C, D, and then of course you've got the vena cava. You have the pulmonary artery. Arteries always go away from the heart. Think of the A on a artery, A for away. So this is the vena cava, pulmonary artery. This is the pulmonary vein, D, and C is the aorta. It's going away. It's a very big artery. Anywho, moving on here, because we don't want to be on too long or keeping it nice, a little bit short. We So your heart, so here's my crusty heart drawing. And I just said, there's your vena cava. That's bringing the blood in, right? Then you got your pulmonary artery taking the blood away. Then you got your pulmonary vein bringing the blood back full of oxygen, ready to go on its mission around the body. And the final route is to come out here. And we're going to go to the whole body, and that's the aorta. Now, what I'm going to do, get rid of that because I just want to concentrate on this, right? So, oi, the way the aorta works, the aorta, right, is going off to the body. It's got to deliver blood to the whole body. This is my crusty little aorta, right? What's the first organ? This is the oxygenated blood, full of oxygen. What's the first organ it needs to deliver blood to? Where's the first destination? What's the closest organ to the aorta? Bit of a trick question. What's the closest organ to the aorta. The aorta is delivering oxygen to the body. What's the closest one? Closest organ is the heart itself. So it's going to drop oxygen off everywhere, but it mustn't forget its own walls need oxygen. So very quickly, a mini artery breaks off of the aorta and delivers oxygen all over the outside, all to the walls. It's branching. This is supposed to look like a tree with no leaves. Yeah, it's like branching away like that. Okay, now this has a special name. This is the first artery that branches off of the aorta. There's what good, thank you. Coronary artery. You're right, coronary artery. Um, there's an artery for your kidneys called the renal artery. So when the blood in the aorta, as it branches off to the kidneys, renal artery. There's one to the liver, the hepatic artery. So there's one to your brain, which comes off and goes up there. You've got the carotid artery. You've got arteries going to all different parts of your body. You've got the femoral artery carrying blood down to your toes and through your leg. But the first artery here is the coronary artery. And this is why we call heart disease coronary heart disease. So the coronary artery is going to supply oxygen to all of those lovely muscles in the heart for the four chambers so they can do their job because they've got to contract. And your heart, remember, is working when you're asleep, when you're unconscious, when you're awake, when you're walking, sitting, studying, it's constantly contracting. So it's a pretty awesome muscle. Um, so yeah, the oxygen comes in here, spreads out here. So coronary heart disease, what happens in coronary heart disease? Well, in coronary heart disease, your artery gets blocked with fat. It gets blocked there, you're dead because um, no oxygen is going to get to your heart and it can't pump blood around your body and you're going to die. What's more likely to happen when you suffer from coronary heart disease is part of it is going to be blocked with fat, and that region is going to die and you will have a heart attack, but you can survive that because it's just part of your heart wall that will no longer contract. It will no longer work. Those cells will die and they probably won't be able to repair themselves very well. But you can change your lifestyle. You can live after a heart attack like that. You can't live after a heart attack. The start of the coronary artery is blocked. That's going to be a massive heart attack, right? But only these cells here will be killed if this one is blocked. Now, um, yes, we have some treatments for this. Ooh, what have I done? We've got some treatments for this. So each organ has different arteries providing them, yeah, with oxygen for So you've got to break it down a little bit more. So each organ has an artery providing them with oxygen so their cells can do respiration to release energy to do all the jobs they need to do. Otherwise, they die. Yes, and the effects on the body, guys, the effects on the body of this are um, how do the cells die here? We have to know how the cells will die here. This is a very important thing that you know the effect, and it's the opposite. So, in fact, I already said it. It's the we already went through it, didn't we? I went through it earlier. So it is less blood flow to cells. What does that mean if there's less blood? It means less oxygen, and you're just using the word less. Less, if you can remember aerobic respiration, but I think they're going to allow you with just respiration. Less respiration is GCSE, and then less energy released. I like to go overboard, right? And that means cells die. So your cell's going to die. If you want to get technical, your cells do have an option to survive. They can try to do anaerobic respiration without oxygen. They're not going to live on anaerobic respiration alone. They will die. But in the process, they'll make lactic acid, which is toxic and causes the pain. So the pain that people get when they have a heart attack, see them grab their chest, that's caused by the lactic acid that's being released as the cells desperately try to stay alive and produce energy anaerobically because they're not being supplied with oxygen. So what are the factors, lifestyle, the ones that we choose about how we live that can affect um, whether our coronary artery gets blocked? So we have lifestyle factors that can affect whether our coronary artery gets blocked. Don't get these confused with genetic factors. You don't have a choice about your gene stuff. Okay. So your lifestyle factors are things like smoking cigarettes, drinking alcohol, eating high fatty, saturated fat diet, um, being overweight, being obese. These are kind of mainly lifestyle factors. I know some genetics can affect some of that. Stress. I choose to be a secondary school teacher and be getting abused by my students all the time, and I'm doing that every day. That's a choice. I know it might be a difficult choice because I've got to earn a living, but that's a choice. Yeah, you're going to be stressed, or if you work in a stressful job. So all of these factors are going to affect um, the chance of you developing coronary heart disease. Genetic factors you can't choose, your ethnicity, your gender, your age, your genetics, basically what you inherit from your parents. You'll know if you have heart disease in your family genetically. You'll know this because you'll know, oh, Uncle so and so had a heart attack, or my granddad died of a heart attack. Oh, Gran had a heart attack. Oh, so and so had a heart problem, and so did. Then you know, oh, it's it running in my family. Doesn't mean you're definitely going to get it, but it means you'd be a mad person if you then decided, if you have heart disease running in your family and then you decide to eat chips and burger every day and never ever walk or do any exercise, and you smoke cigarettes and you live a stressful life, then you're basically painting a bullseye on your head for a heart attack. But yeah, so you have to be aware of lifestyle factors, genetic factors, things like that. Yeah, foods high in cholesterol. Now, there's some cholesterol is good and will unblock your heart, so to speak. It's a bit more complicated than that, but it will store the fat not in your blood. It will take the lipids and it won't go in your blood and block your coronary artery. And there are certain cholesterol that's bad for you, and we call them HDLs and LDLs, high density lipoproteins and low density lipoproteins. HDLs, P density lipoproteins, and LDLs, loser density lipoproteins. Um, LDLs are the ones that will cause fat to build up in your coronary arteries, and they're found in animal products. So any animal products are very high in LDLs, and plant foods are high generally in HDLs. Yeah, and exercise is key. Right. So if you do develop heart disease, there are two treatments, and you could get a fat, nasty as Eshel said, you could get a fat six marker on them. So this is what happens when you have coronary heart disease. Fatty deposits build up in the wall of the artery. It doesn't quite look like this. They actually build up inside the wall and they make the wall bulge. So the wall comes in like that. Now we have an option. You can get something to push it back open, and that's an operation. What they're doing here, they're inserting a stent, which is like a metal cage, into the artery, and this will hold the artery open. Um, you need to know about the downsides to that and the good news. The good news for both is exactly the same. See what I said here? Less blood flow to cells, less oxygen, less respiration, less energy released. We just change all of them. So if they say, well, how do statins help you? What is more blood flow? More oxygen, more respiration, more energy. So how do stents help you? Well, they increase the blood flow. So you get more oxygen to heart cells, more respiration. So you get the idea, right? You just reverse the issue when you're writing your answer. Now, you need to know some verses like, okay, so which one is better? Well, it depends on the patient. This one, you get treated and you're done. This one, you got to take pills every day for the rest of your life, and you've got to remember to take the pills. Um, Pills, medicines come with side effects. Headaches, it could be just headaches. But it could even be organ failure, like, and death. So some people, these may kill. Okay, very small amount. There's a very small risk of like fatality, organ failure, might damage your liver or something like that. So, very small risk. With this, you could get an infection. You're not going to get an infection from taking medicine, but you could get an infection. Anytime they cut you open, bacteria and viruses can get in. You could get an infection at the surgery site. So, yeah, you need to know for and against for statins. So both of them do the same job, which is increase the flow of blood, increase the oxygen to cells, increase respiration, more energy in the cells. Okay. So they're both going to make you feel better and healthier and reduce the chance of a heart attack. Um, Statins, you've got to take every day. Statins have side effects. Could be simple, mild ones like headaches or something, all the way to organ failure. And these, you could get an infection. Well, and also stents. Anytime we put something in the body that's artificial, that's not made from living tissue, you can get something called a blood clot. Wait, so we can get like a blood clot, and that in itself can give you a heart attack. So in a way, the stent could cause you to get a blood clot, which could lead to a heart attack. You might say, well, how's that? Well, take the smoothest mirror you have. See my lovely coffee cup? The inside is very smooth of my coffee cup. But if you zoomed in on that, it's actually a bumpy, rocky surface in comparison to the inside of your artery. Your artery is so smooth, we can't make things as smooth as that. So even a piece of metal is bumpy and the blood can stick to it. Even a smooth piece of metal, we can't mimic how smooth our stuff is. Like it's designed so that blood doesn't stick to it. Your natural arteries have evolved that way. Okay. What do we want to do? Oh my good gosh. Right. Let's move on. Let's move on. I'm taking too long. Should be just a little bit more, about 10 minutes more, and we'll finish this topic for the moment. So make sure you know what these are, A, B, C, and D. And again, you can pause, you can watch this back, pause it, make a revision card on any of these. You make the question up yourself. The beautiful thing when you make revision cards, I'm a big fan of revision cards, obviously, but the beautiful thing when you make revision cards is you get like what you revise in a certain way when you make the card. So when you make the card, you're forced to think of a question. So just having to think of a question is helping you because you're like, all right, I'm going to draw out these and say, what is each of these? And then on the back, you put the answers. You've had to think of a question and you've had to think of the answers, and you can check them out in revision guides or online. So you've got red blood cells. Now, a lot of students would be like, oh, but that's a white blood cell, sir. What they're trying to show you here is that donut concave shape. Red blood cells have a shape like a donut, and it's so that they have a large surface area for gas exchange to pick up oxygen. They have no nucleus and they're filled with a chemical called hemoglobin. Now, actually, B is a white blood cell. Now, how would you know that from looking at the diagram? White blood cells have a nucleus. Look at this giant fat nucleus these guys have got. Okay. So, white blood cells have a nucleus. Sometimes, if they make antibodies, they just have a smooth round nucleus. If they're phagocytes and they engulf and digest, they have like a weird lobe nucleus. And then we've got C would be plasma, is the yellow liquid the blood floats in. And D will be platelets, which are broken bits of cells. Yeah, well done. No problem. Keep up the good work. Okay, cool, cool, cool. Coolio, 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 coolio. Um, have a good time, have a good time. Right. So this is the functions. So you'll need to know the functions of your blood components. He says there's a couple more slides to go through, then we're done. Because I've got to go through transpiration and translocation as well, and then we're done. No problem. I'm not good at spelling either. Right, name the part of the blood. So, name of the part of the blood and the function. What do red blood cells do? What do platelets do? What does plasma do? What do white blood cells do? And so, what we want to do here, red blood cells, you can do this in your head and see which one you think it is. I'm just going to smash the answers on because I take too long otherwise. It's the problem. When I try to revise the night before the exam, it ends up taking 4 hours and it kills me. So, red blood cells transport oxygen around the body. Platelets clot the blood. White blood cells fight disease or they kill pathogens like bacteria, viruses, fungi, and protists. And plasma carries, ooh, yeah, plasma carries dissolved hormones. Hmm. I don't like this answer. I don't like this. This is a crap question because carries dissolved urea. Well, anyway, I'm not going to get into that. It carries dissolved hormones. So the plasma has loads of dissolved stuff. It has urea, hormones. It's a bit of a silly one because they should accept plasma carries dissolved urea as well. That's a bit rubbish, that. But never mind. Right, couple of things. I need to move on because I'm just wasting time. Right, there's something called artificial blood. I haven't really ever asked a question on artificial blood. It is on the specification, but they've not really asked a question yet on artificial blood, so I wouldn't over worry about it. Artificial blood, just the main reason for artificial blood, when we have a lot of blood loss, say during a car accident, if I'm bleeding and I'm losing a lot of blood, the main issue is not really the oxygen carrying capacity. It would be ideal, but is really that my blood pressure drops, and it's your blood pressure dropping and your blood vessels collapsing, and basically it's the blood pressure dropping too much that's bad. So even if I just pump you with water to replace the blood, it will keep your blood pressure up and keep you alive until we can get to the hospital. So if I could put water directly into your veins and fill your system with water at the same rate you're losing blood, I can keep you alive and we can get to the hospital. Now, ideally, artificial blood shouldn't just be water. It should have salts in it, it should have sugar in it, and all the normal things your blood carries. We haven't found something that can pick up and do the job of a red blood cell yet and carry oxygen, but they are researching, and there are some people trying to find that so they can make a really good artificial blood that will also be able to do the job of the red blood cells. You could literally survive on just like a quarter of your red blood cells, even if you lost a lot of blood. You'd have enough if you're not running around because you're lying down on a stretcher. You've got enough red blood cells to keep you alive. It's really the blood pressure dropping that's the problem. All right, let's zing to. Oh yeah, antibody production with the mouse. That's going to be in like my fourth biology one when I do unit 4. That's in immunity. Yeah, when we talk about the hybridoma and fusing the tumor cell with the B lymphocyte from the mouse. Now, cancer, this comes up in AQA Biology Unit 2. And what do you need to know about cancer? Cancer is uncontrollable cell division. So it's when a cell divides uncontrollably. Now, there are two types of tumor that you can get. One is not a cancer and one is a cancer. Do you know the name? Um, yeah, so we have artificial blood at the moment, but it could save even more lives if it could carry oxygen. At the moment, we just give you like a salt water solution to replace your blood until we can get you to hospital. And then if you've lost enough, you can have blood transfusion, extra blood put in you from somebody else that they keep in the freezer at the hospital. Now, um, cancer can be benign or malignant. Well done, excellente. So you have to know the difference between benign and malignant. And here's an example of a question. Malignant tumors are cancers. 7.1, describe what happens to cells when a tumor forms. So when a tumor forms, cells do cell division uncontrollably. They grow uncontrollably or divide uncontrollably. So instead of going like divide, divide, and you just get this big ball of cells, which of course is bad. And the evidence that this is malignant is malignant tumors spread from one organ to another. So if you find you've got cancer in your lung, spread to your back, your spine, your blood, it can spread everywhere. And they travel through the blood and they spread to different organs in the body. They travel through something called your lymphatic system as well. So they can travel through your lymph, which is another fluid you have in your body and another system, or they can travel in your blood. So the main thing with malignant tumors is that they can spread and invade organs and they travel in the blood. Not benign, good tumors. I mean, no tumor's good, but a benign tumor, it doesn't spread and it doesn't invade organs. It's kept in a capsule or a membrane, not a cell membrane, but a membrane. So, like, if this would be a good, when I say good, I shouldn't say good. This would be a tumor that is benign. So it's a ball of cells, a bit like an embryo. It's a ball of cells, but it's kept inside a capsule. It's trapped inside a kind of bubble, so it cannot invade your lungs, your liver, and it go and travel. Um, a malignant, dangerous tumor is not in a capsule. It's not in a bubble. So it can break off and they can travel around your body through your blood and invade and spread to other organs. So they are bad. So you have benign and you have malignant. Cool. And if in doubt, just ask Google. Look, I just asked look, I can't even spell right. Look at that. I called it malignant. It's malignant. I missed out an N there. Anywho, look, AI overview. Check it out. I said, according to AQA, I just put in malignant versus benign tumors, AQA biology. So these are the type of things. Use your internet, use your tools. According to AQA biology, malignant tumors are cancers. They grow rapidly. They spread via the blood and invade organs, tissues, etc. Benign tumors are not cancers. They grow more slowly. I mean, they still grow rapid, but they grow more slowly and they're within a capsule or a membrane. I know you can say capsule if you prefer that, or a membrane. And they remain local. They don't go to other parts of the body and they don't travel in the blood. Cool. You're right, Ishul. You should really say cell division. So they grow by so uncontrollable cell division or uncontrollable mitosis, if you prefer. Uncontrollable mitosis would be a shorter one. Right, the final thing I'm just going to finish with because I've gone on a longer than I wanted to is xylem versus phloem. Xylem is a tissue, not an organ, but tissue, and the phloem is a tissue. So a group of similar cells doing the same job. Now the xylem's job is to transport water and mineral ions around the plant. The job of the phloem, and I think food. See, it says food. So now small food molecules, it transports them. So you could be asked, 6 marker, what is the difference between the xylem and phloem? So let me smash through the differences. Yeah, I'll hit transpiration and translocation. I'll wrap that in this as well. So xylem and phloem, you're right. Transpiration is in the xylem and translocation in the phloem. Not really. The xylem isn't really transpiration. At GCSE, we can probably say it's transpiration. Really, the xylem is part of the transpiration stream. Transpiration is the evaporation of water from the stomata in the leaves, but it starts with water going up the xylem. So if I'm comparing the two, I want to say the xylem is made of dead cells. The phloem is made of. So if you're asked to compare, there's two ways you can do, depending on how you like it, right? This is your choice. If you get a 6 marker and you're comparing two things, you can go, you can go piece for piece. Some people like that, or some people just like, right, I'm going to write about the xylem, and then I'm going to write about the phloem. But some people prefer to go, which is the more complicated, more difficult skill, probably examiners might like it better, is where you do a direct comparison. So you're like, the xylem is made of dead cells, the phloem is made of living cells. The xylem has lignin in the walls for extra strength. Remember, it's transporting water, it could explode and burst. So it doesn't just have cellulose, it has lignin. They both have cellulose in the walls. They're both made of plant cells. So the xylem has lignin, no lignin in the phloem. The xylem transports water and minerals. Phloem transports small food molecules. If you want to be precise, you could say amino acids and sugars. The xylem goes in one direction. The phloem goes in two directions. The xylem uses transpiration. The phloem uses translocation. So you see, you can do direct like that, or you can just go, I ain't doing that. That's going to mess me up. I can just write about the phloem. Sorry, write about the xylem, write about the phloem. You can do it like that. Um, no end walls. So what do I mean by no end walls? Imagine these were once plant cells like this, right? These were once plant cells. And what happened is the plant went, right, I'm going to make a xylem here. So it carved away the horizontal end walls to just leave dead cells. It rotted through the cytoplasm. It burnt it all away, got rid of the nucleus, got rid of everything. And the idea was to create a tube. So there's no walls at the end of each cell unit. They've been smashed away. And that's to create a low resistance. So you make a pipe where the water can just flow. You don't want cytoplasm or a nucleus in the way. The water's going to hit it and slow down. So it wants a nice clear pipe. The phloem actually has plates. You can just say the cell wall at the end has holes in it. You don't like, you can say holes in the end walls. It's actually the technical name is sieve plates because they look like a sieve, like when you're watching rice or pasta or draining rice or pasta or something. They look like a sieve. So yeah, and they go in both directions. So there's your direct comparison. Now, Any questions about any of that? You just got to smash that on a revision card. Again, your revision card could be compare the xylem to the phloem, or you can make two revision cards and have one like describe the structure and function of the xylem, describe the structure and function of the phloem, or you can just put it all on one revision card and on one side you write the question, describe, compare the phloem and xylem. What should happen then is by the time you get to the exam, you're bussing through, saying the question and you're saying the answer, and then you go check the back and you're like, yeah, I got 90% of that. I'm good. So, um, the xylem is a passive process. The water is like sucked up. The phloem uses active transport at a certain stage. You don't need to know the details of that, but there's some energy used in this process. I'll be doing physics and chem as well. Yeah. Yeah, I'll be doing physics and chem as well. And the night before the exam, I do like a 3, 4 hour one, but I will do some physics and chem. I might swap to chem next. I might stop the bio for a bit. I've done B1, B2. Well, when I say B1, B2, I've done the first 2 units of the bio. Couple of other things. Make sure you know the names of all of this. I'm going to finish in a minute, so I'm not going to go through all of this massively, but waxy cuticle. Then you have these colorless transparent cells, which are known as the upper epidermis, and they allow light to go through. The light needs to go here. These are palisade cells. They absorb light for photosynthesis, hence they're packed with chloroplasts and chlorophyll, the green pigment for absorbing light. And then look, there's our friend the phloem and the xylem, known as the vascular bundle. Do you need to know that? No. But the water can come up and get in there. That's why it's being brought up the xylem. The water is being brought so it can meet the sunlight and make sugar. Why do we have a phloem in the leaf? Why does the phloem go all the way to the leaf? Simples. We want the sugar that's made to go back down through the phloem down to the roots where it can be turned into onions and carrots and potatoes and be stored as starch in the roots. So you've got water, and then of course we've got the carbon dioxide that's coming in here by diffusion through the holes called stomata. And one of the unintended consequences is that water evaporates by transpiration from here. We want the water here. Unfortunately, if we're going to let carbon dioxide in, water's going to escape. There ain't a lot we can do about that. So that's a consequence of having to open the holes called the stomata to allow carbon dioxide in. Remember, stomata are not cells, they're holes. The guard cells are cells. Yeah. And then you have this spongy mesophyll, which is for gas exchange and has air spaces to allow the fusion of gases. Now, it's kind of both triple and combined. I've done both. I'm going to go through the core practicals at some point for B1, paper one, and then I'll do C1 and physics and that as well. I just haven't started on them. Remember, your biology's first, chemistry C1's will be a few days later. Physics, you got an extra weekend normally. Physics always happens after a weekend. You get an extra few days for your physics P1. And then B2, C2, P2 are after the half term at the end of May. They're at the start of June. Always after the half term, you get a full week where you can forget everything you learned in the ones and be like, I'm just focused on the twos. P2's probably is P2's the exam where people do the least revision because some of your friends have finished their GCSEs and you're burnt out. You've done 15, 16 exams by the time P2 comes late in June, mid-June, P2 is. And by that time, people are just burnt out and they're like, I ain't revising. Your friends are going to the park and sunbathing and chilling out. And a lot of people just stop, give up before P2. So you've got to stay strong for P2. Anywho, what are we doing here? Yeah, make sure you know this as well. This has caught some students out, and then I am finishing. I will go over some. I have missed out some bits. Make sure you know the difference. You can put this on a card. I'd put this on a revision card. This comes up every now and again. Make sure you know the difference between the two. These two things are different. Transpiration and the transpiration stream are different. The transpiration stream is 3 marks. Transpiration is 1. Transpiration is this, the evaporation of water from the leaf. So if I had a revision card, what's the difference between transpiration and transpiration stream? I'd be like, transpiration is evaporation of water through the stomata in the leaf. So that's like kind of 2 bullet points, evaporation and stomata, water. Water evaporates through stomata. That's transpiration. Transpiration stream describes how water moves into the roots by osmosis. It's carried up the xylem in the stem to the leaf and then evaporates from the leaf. So it's like three things. It's the journey of water from the roots to the stem to the leaf and out. That's called the stream. So the xylem is really part of the transpiration stream. It's not really transpiration. But transpiration is just the word for the evaporation of water through the stomata. Anywho, I'm going to finish there, guys. Don't forget sharing's caring and all that business. Hit the like button, tap the bell, subscribe. Make sure you've subscribed. You can check out my walkthroughs. I will put some links to some of my specific walkthroughs for the B1 exam and etc. Leave any comments about any suggestions or anything you'd like to go over. Didn't really go over everything today. I didn't mention the stomata and how the shape of the guard cells helps them open and close. Basically, you can see the inner wall is fatter than the outer wall. And this means when water goes into the guard cell, um, when the guard cell absorbs lots of water, the fact that the inner walls are fatter than the outer walls means the guard cell open the stomata. Okay, when they become turgid, they open the stomata. When there's not a lot of water about, it's probably a hot day, the guard cell shrivels up and becomes flaccid or plasmolyzed, and basically that closes the stomata, preventing more water loss from the leaf. Anywho, I'm gonna finish there. I'll catch you a lot later. So take care and goodbye. Um, cool, nice one. And yeah, same time tomorrow, 4 o'clock. I'll be covering a different GCSE unit. So yeah, take care, guys. Nice one.